Hello, my name is Christine Cooper.
I'm Ayali Soria.
And I am Wasim Musleh.
We will be presenting on antagonistic versus synergistic antibiotic interaction.
In synergistic interactions, bacteria can be subjected to the use of multiple antibiotics where there are typically two different types of interactions that occur, direct and indirect interactions. Direct or physiological synergistic interactions cause both of the antibiotics to be used to essentially provoke the effect of the other antibiotic. Through provoking the antibiotics' effects of one another, it causes mutations to occur where a resistance can be formed. However, it also is able to alleviate and remove the synergistic effects of both the antibiotics that are used. Interactions that are considered indirect are also known as epistatic interactions. So in synergistic epistasis, the overall cost of fitness in both of the antibiotics are used in order to reduce the overall amount of resistance formed. Since in synergistic interactions the antibiotics are able to aggravate the effects of the other antibiotics, it can lead to mutations of resistance that are associated to a large amount of benefits. This is due to how the resistance mutation is able to alleviate the inhibitory effect of one antibiotic while simultaneously removing the combination of the multiple antibiotic synergistic effects. This process is able to skew the DFBM, which stands for the distribution of fitness effects of mutations, towards large effect mutations, which lead to rapid adaptations.
Antagonistic interactions between antibiotics essentially just aggravate each other's effects, and the effect of one antibiotic will compromise the effects of the other antibiotic. This will in turn lead to the marginal benefit. When acquiring resistance to one antibiotic, it makes an unmasking of the inhibitory effect of the second antibiotic. This results in a DFPM skewed towards mutations containing small effects, leading to a low rate of evolution in multi-drug resistance with antagonistic epistasis. This typically occurs between resistant mutations that target different steps of the evolutionary pathway, as we as well as supporting the connection between antibiotic resistance and genetics and physiological manipulations of both gene expression and protein synthesis.
In box 2, it represents the DFBM, or the distribution of fitness effects of beneficial mutations. In sections A and B, it shows a hypothetical fitness distribution when there's a low selection of spontaneous mutations. Conversely, sections C and D show a hypothetical fitness distribution when there is a high selection of spontaneous mutations. The blue shaded area of each of the four sections represents the fitness of the resistance mutations, and the dashed line represents the wild type fitness of beneficial mutations. In sections A and B, we can see that the DFPM is considered to be exponential, where in sections C and D, it is no longer exponential. In box 3, it depicts the fitness interactions of genetic interactions as well as drug-drug interactions. Essentially, when there are multiple drug-resistant mutations of a strain of bacteria, the cost of resistance is dependent upon the individual resistance mutation cost, as well as how the interactions that are epistatic of the mutations that are resistant. In section A, we can see how interactions that are additive indicate that the effects of the mutations are independent of the overall fitness. Interactions involving synergistic epistasis deal with how the mutations provoke one another's cost, and that antagonistic relieve the cost of the mutations. In section B, antagonistic interactions compromise the effects of antibiotics, and synergistic interactions promote the effects of the antibiotics, and additivity occurs when each of the antibiotics are independent of one another.
The usage of the over the counter antimicrobials can result in ineffective in the medication for the person's future health. Penicillin is an example of an antimicrobial that has threatened many illnesses such as pneumonia, meningitis, scarlet fever, blood poisoning, strep throat, and gonorrhea. But the overusage or the lack of treatment can cause bacteria to grow resistant to penicillin. Common cold and other minor sicknesses can increase the ineffectiveness of these antimicrobials and make it extremely difficult to treat greater illnesses through the road. But there is still a large debate, and putting society first seems to come first at all costs, and the increase of anti resistance continues to grow. The author questions whether the usage of antimicrobial resistance on injuries is a tragedy of commons in figure 2. There is a different trend line wherein tragedy does not appear. They share a common optimum. The tragedy occurs when there is an individual competition over the resource, and then in return, there is a reduction for the overall fitness of the entire group.
Drugs, like genetic perturbations, can have synergistic, additive, or antagonistic effects or interactions. Antagonistic interactions have begun to be appreciated for their ability to uncover gene and drug functions, but they are generally avoided in the clinical world of antimicrobials. Synergistic drug combinations generate increased efficacy at lower doses, aka the minimum inhibitory concentration, which is why it has long been used by clinicians. Suppressive drug combinations have been shown to reduce inhibition power at fixed drug dosage, but their impact on the evolution of drug resistance has received little attention until the past few years. As we know, it is becoming increasingly more difficult to make new antibiotics and continue this fight or arms race against bacteria who are becoming more resistant to our antibiotics and arsenal. For environments involving a single drug, a key factor that affects the propensity for evolution of resistance is the frequency of resistant mutations as a function of drug dosage. The drug concentration that corresponds to the final step at which the frequency of resistant mutations drop below a detectable level is termed the mutant prevention concentration, MPC. As evolution requires both mutations and selection, resistance is thought to evolve primarily at drug concentrations below the prevention concentration but above the minimum inhibitory concentration, a region known as the immune selection window. As drug concentrations fluctuate in time and space in and out of the immune selection window, the size of the immune selection window is thought to influence the chance that resistant bacteria will evolve. It is worth noting, however, that in addition to the time within the immune selection window, other factors such as the time spent at low and high drug concentrations within the immune selection window must also be considered. A few studies examined the mutant selection window in several drug pairs and demonstrated that although some drug combinations can substantially narrow the immune selection window, others cannot. To understand the impact of drug combinations and of the interaction between drugs on the size of the immune selection window, we need to consider a model that can predict the number of resistant colonies appearing in a multi-drug environment. In the model created by the researchers, they incorporated drug interactions and cross-resistance, making the assumption that each mutant experiences the same interactions as the wild type. That is, resistant mutants see an effective reduction in drug concentrations, and the region of growth in the two drug space can be approximated by rescaling the region of growth measured for the wild type. This assumption yields a model that predicts the frequency of mutations, allowing growth in multi-drug environments based on the profile of resistance to each of the single drugs as a function of drug dosage, the level of cross-resistance between the drugs, and the type and extent of their interaction. Model predictions agreed with high throughput measurements of resistance frequencies over the two-dimensional drug dose space for several different drug pairs. Importantly, this model suggests that for fixed levels of cross-resistance, antagonistic drugs decrease the size of the mutant selection window more than synergistic interactions. In this figure, Part A shows a schematic of the frequency of spontaneous resistance mutations as a function of drug concentrations for a single drug environment. The point at which the frequency drops to undetectable values is the mutant prevention concentration. As evolution requires both mutations and selection, evolution is thought to proceed primarily below the mutant prevention concentration, and above the minimum inhibitory concentration, this region once again known as the immune selection window. In part B, we can see that the concept of the immune selection window can be extended to multi-drug combinations, for which it becomes the area red, which is between the minimum inhibitory concentration lines shown in solid lines and the minimum prevention concentration lines shown in dashed in the drug-drug concentration space. A fixed drug ratio corresponds to a linear line with angle theta that extends from the origin and is associated with an effective mutant selection window. And so, in their experiment, at drug concentrations below the mutant prevention concentration, resistance mutants will appear. Mutations that confer resistance to a single drug may be more beneficial for bacteria in multi drug synergistic treatments than in antagonistic treatments. With synergistic drugs, single drug resistance mutations not only remove the effect associated with one of the drugs, but also remove the synergy with the other drug. With antagonistic drugs, by contrast, removing the effect of one of the drugs will actually reveal the previously suppressed effect of the other drug. These expected differences in the fitness advantage that resistant mutations confer in multi-drug environments suggest that the rate of adaptation of the population may actually be faster in synergistic drug environments. A study designed to test this prediction has used automated parallel evolutionary adaptation experiments, in which the bacterial populations are propagated in different drug combinations over hundreds of generations while their fitness increase is monitored continuously. Results of this study, as shown in Part B, illustrates that synergy accelerates the rate of adaptation relative to antagonistic drug combinations.
Article 3 describes a phenomenon called the tragedy of the commons, describing how the overuse of antimicrobial drugs over time leads to the inability of the drug to be effective as a treatment. This relates to the outside article due to how it depicts how antagonistic and synergistic interactions occur from the use of multi-drug treatments. An example of this includes studying doxycycline resistance in bacteria resistant to doxycycline. The resistance to doxycycline in bacteria that are resistant was able to outcompete the wild type that is sensitive to doxycycline. However, when observing doxycycline with its synergistic counterpart of erythromycin, the bacteria resistant to doxycycline is able to remove the resistance to both doxycycline as well as the erythromycin. Through using ciprofloxacin, the doxycycline suppresses the ciprofloxacin, which in turn causes the wild type that is normally sensitive to doxycycline to outcompete the bacteria that is not sensitive to doxycycline. By using multiple types of antimicrobial drugs as a form of treatment, it could potentially lead to a tragedy of the commons. As with more and more people using these antibiotics, it would eventually create health costs through selecting from microbes with more resistant strains.
The first paper from Module 2, the author talks about antibiotic resistance. One thing that we have to keep in mind is that bacteria reproduces very fast, making it difficult to stop the antimicrobial resistant genes from reproducing. They also mentioned that the idea of multi-drug treatment, this is where the topic arises of antagonism is brought up. The antibiotic resistant is very similar to the antimicrobial resistant that we see in paper 3. There is a trend of fitness growth rate in paper 2, and in paper 3 there's a trend how when antimicrobials can reduce or the lack thereof antimicrobials can either weaken or have no effect when it comes to treating infections later on.
The choice between synergy drug interactions versus antagonistic interaction involves a trade off. And we need to ask ourselves, should humanity focus on the immediate efficacy of inhibition of microbial growth or stop giving ill patients antibiotics to slow the evolution of these microbes?
This is our work site. Thank you for watching and have a great one.